Good afternoon, everyone. My name is Ann Patchman. I'm a member of TIARA, and I would like to welcome you to Saturday Session 33. I told Dr. Gleeson I'm going to start with a joke. I used to say I was 100% Irish, and then I had my DNA done. I'm only 91, and what really irks me is my sister, who thinks all of this genealogy stuff is rubbish. You know, she keeps saying to me, "Are they still dead?" Is 95% Irish. She's more than me. So, told you I was going to start with a joke. So I'd like to introduce Maurice Gleeson, who is a psychiatrist and pharmaceutical physician, as well as a genetic genealogist. He is administrator of several Surname DNA projects, works with adoptees, and has appeared on TV as a DNA consultant. He authors several blogs, is a regular contributor to genealogical magazines, and his YouTube videos are very popular. He has organized the DNA lectures for Genetic Genealogy Ireland in Dublin and Who Do You Think You Are in the UK since 2012, as well as given talks all over Ireland, the UK, and internationally. He was voted Genetic Genealogist of the Year 2015 from Surname DNA Journal, and from Canada's Anglo-Celtic Connections, he was voted Superstar Genealogist Ireland. So please welcome Dr. Leeson. Thank you.
Thanks very much. Thanks very, very much. Well, today I'm going to talk to you about using YDNA to research your surname. How many people here have actually done a YDNA test, or have got their brother or a male relative to do a YDNA test? Great, so a lot of people, okay. One of the big things that people say when they get their YDNA results is, okay, well, what does this mean and what do I do with them? So hopefully by the end of this lecture, you'll have an idea of what kind of projects you can join, what your YDNA results actually mean, and you'll get an idea of the bigger picture of the whole field of YDNA. Now, I've got a lot of slides. I'm recording this for you, so you'll be able to see this again on the website, so you don't need to be furiously taking notes. It means that you can watch the video again and again should you so desire, and it just makes things easier for you, and you can relax and try and let it wash over you. Some of it will stay inside, some of it will go out the other ear. Don't worry about that too much. It does take a while for this DNA stuff to sink in.
Does all of this change our structure just listening to you?
Just listening to me, it's going to filter by osmosis from an area of high concentration into an area of low concentration. So that's how I learned it. So let's hope for the best. So there are three main types of DNA, but the YDNA goes back along the father line, the direct male line. And it has a reach of about 200,000 years, so it's very, very useful for migration studies. And the same with the mitochondrial DNA on the other side, female, mother line, again, very useful for migration studies. And the third type of DNA is the autosomal DNA, which is the one that we mainly use for genealogical purposes, but it only goes back about 300 years. So it's going to connect you with genetic cousins with whom you share a common ancestor sometime from about 1700 onwards, sometimes within that time period. But to get back beyond that 1700 time point, the YDNA can be very, very useful if you are researching a particular ancestral line. Now, it could be your surname, it could be the surname somewhere in the middle of your family tree, in which case you have to find a male cousin with that particular surname and get them to do the test for you and be your proxy for that particular male ancestor in the middle of your family tree. But if it is your surname, then it's going to be on your father line, your direct male line. So YDNA and mitochondrial DNA useful for deep as well as recent ancestry, and autosomal DNA only useful for recent ancestry from about 1700 onwards. So YDNA, let's look at this now. It tells you about your deep ancestry, but it also connects you to surname projects. How many people have started their own surname project in the room? 1, 2. Okay, there's 2, and I've got about 10, so that's 12 of us. So,
Could you define Surname Project?
A Surname Project is a project that uses YDNA to research a surname, a specific surname. So my surname is Gleeson. I've got 170 Gleesons in my surname project, and it divides the Gleesons into at least seven now at this stage, 7 distinct genetic groups. Some of them came from Cork, some of them came from Tipperary. So a surname project is a way of looking at your surname, how many different genetic groups there are within that surname and where they all came from. And you'll see from a few of the examples that I've been able to link some of the Irish surnames I work with back to the ancient annals, so that people in those surnames, in those specific genetic groups, in certain surname projects, can actually link their genealogy reasonably confidently back to the ancient annals and go all the way back 1,000 years to the person who first started the surname. And that's really quite amazing. It's also a great way of engaging the local Irish into doing a DNA test, because trying to get DNA out of an Irishman is like trying to get blood out of a stone. So it's a great way of actually engaging the local Irish in doing DNA testing. Question here.
I didn't understand one of your words. The ancient annals?
The ancient annals, or the ancient historical texts. Annals are spelled A-N-N-A-L-S. So an annal is like an ancient tome, an ancient book, and it contains the history of a particular line of descent, going back to, say, Brian Boru or Niall of the Nine Hostages, one of these ancient historical figures that maybe gave rise to a whole group of surnames, not just the O'Neills and the O'Briens, but a lot of related surnames. And I'll show you an example of one of those later on, because some of them are available online. So you can actually, they've been transcribed and put up online, so you can see the line of descent going all the way back about 1,000 years or so. So when you get your YDNA results on Family Tree DNA, they're the only company that do YDNA testing that's useful for surname studies, this is what you'll get on your home page. You have, there's my name up there, there's my kit number, it tells me my haplogroup is haplogroup R Y16880. I'll explain what that means in a while. There are my details there. And on the actual page itself, you want to click on your matches to see who else you match on your YDNA. And also you want to see the haplotree and the SNPs. Now the haplotree is just a name for the tree of mankind. If you imagine that genetic Adam was some anatomically modern human that arose about 250,000 years ago in Africa, and he has actually given rise to all modern humans today. It's not that there was just one person at that particular time, it's just that the descendants of only one of those people have survived to the present day. There were lots of other men around at that time, but all of their descendants have died out. In fact, 95% of ancestral lines have become extinct over the passage of time. And we are really just a 5% snapshot of what could have been, had descendants survived to the present day. So if I click on matches, then you'll see that with my particular YDNA at the 67 marker level, I have 8 matches: Little, Gleeson, McLaughlin, Gleeson, 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 McLaughlin. So quite a few of them are called Gleesons, which makes me think that I'm probably a Gleeson, which is probably fairly good. It means that there hasn't been a surname or a DNA switch somewhere along my father line, going back along maybe 1,000 years back to the person who originated that surname. Other points to note here are the most distant ancestor is the most important piece of information that you can include in your profile. So when you do the test on Family Tree DNA, you have to fill in your own profile, and they ask you for your most distant known ancestor on your father side and your mother side. For the YDNA, the father side is the most important. Put in as much information about his birth location as you possibly can, because that's going to be very, very important for helping yourself and other people to figure out where your own particular genetic group came from. Now, other things to note here would be the fact that you can test 37 markers on your Y chromosome, or 67 markers, or 111 markers. So there are 3 types of tests, and we'll look at those. Here is an example of what a surname project looks like, and I showed this yesterday. It's simply just to give you an example of what the final form is going to look at. So if you just go back 1 slide to your matches, I have 8 matches here, and it doesn't tell me a huge amount about what my YDNA actually means. It's only when you compare your DNA to other people's DNA in the database that you start getting lots of useful information. And that's why it's important to join a surname project. And there are over 9,000, actually there's over more than 10,000 surname projects on Family Tree DNA at the moment. How many people have joined a surname project on Family Tree DNA? Quite a few people, okay?
Would you be talking a little bit about genetic distance?
Yes, a little bit. I mean, I can talk about that now. This column here, genetic distance, you can see these numbers 2, 3, 6, 7, 7, 7. It's basically the number of steps away from an exact match with each of these people here. So if you look at the first column there, it says the genetic distance is 2 to this person called Little. It means that I am 2 steps away from an exact match with this particular individual. If it was an exact match, then it would be 0 out of 37, or in this case, 67. 0 out of 67.
I have a 0 on the
Then you have an exact match then. If you have a 0 out of whatever level of comparison you're using, 37 markers, 67 markers, 111 markers, then that is an exact match. That suggests you are a closer match than somebody who is, say, 7 out of 67. So it's an indication of how closely you match your matches on the matches page.
Just one more question.
Sure.
And 111 would be what percentage of the total steps, or whatever, in YDNA?
Well, you can have 111 markers. So a 0 out of 111 is obviously going to be a very, very close match. And they reckon that 50% of people who have a match at 0 out of 111 are first cousins or closer. So if you get a match at 0 out of 111, there's a 50-50 chance it's a first cousin or closer than that, which is very useful if you're a male adoptee and you do the YDNA test and your result comes back and it shows that you are 0 out of 111 to this unknown person that you've never heard of before. There's a 50-50 chance that they are either a first cousin or an uncle or a half-sibling. So it can be very, very useful for adoptees to do this type of test. Also, if I was an adoptee and I got these results, as an adoptee, I wouldn't know who my father was, who my biological father was, but just looking at these results, there are 1, 2, 3, 4, 5 people up there called Gleeson. That's a very, very good indication that my biological father was probably called Gleeson. And it helps me narrow down the search for my biological father because genetically I'm related to 5 Gleesons there. So it's a very, very useful test for adoptees as well. But once you have your results, you should join surname projects, among others, and we'll talk about them as well. And in a surname project, what they do is they list people. Here's a list of the people in this particular genetic group. And imagine that their Y chromosomes are stacked up on top of each other. And the various markers along the Y chromosome are here at the top. The most important thing to look at in this slide are the pretty colors. Because the pretty colors tell you that there is a pattern. Can you see a pattern there, a colored pattern in these groups? Here's group 1, and these bars, these colored bars, go down along almost the entire length of the column in most of these places. But this colored pattern that you see here in group 1 is very different from the colored pattern you see in group 2. And we also have a third group, and you can see that the colored bars are very, very different from the colored pattern in the other 2 groups. So what you're seeing here is 3 groups of people that have their own unique colored pattern, their own unique genetic signature. So this is what I love about this particular website, worldfamilies.net, which unfortunately no longer exists because it was pulled down because of GDPR. and the new European law, but it did allow you to very easily, just at a glance, see what the genetic signature is of a particular group. And in group number 2, lineage 2, we can say anyone who matches this group and has this colored pattern, they come from North Tipperary. Anyone who matches lineage 3, which is this one here, that colored pattern comes from West Clare. And anyone who comes from lineage 1 goes back to Cockfield, Suffolk in England, but not only that, goes back to a specific named ancestor, born in 1609. And of course, a lot of people will be brick-walled at about the 1800 time point. So to be able to go back to a specific named ancestor is like having your genealogy handed to you on a platter. Now, there's going to be maybe 3, 4, or 5 generation gap between where you're brick-walled in your YDNA line and the actual ancestor, Thomas Gleeson, born 1609. But at least the YDNA is pointing you in the right direction. It is helping you focus your genealogical paper-based research. Whereas previously you were brick-walled, now you have a target to aim for. And there's other groups within this particular project that we don't know where they came from. And then there's also a large group of ungrouped people who may be rare Gleesons, and they're the only Gleeson in that particular rare genetic group to have tested so far. They have to wait in the ungrouped section until more people join the database, and then hopefully somebody else from their rare branch of the Gleesons will join the database and will be a match, and then the project administrator will take them out of the ungrouped section, and they'll put them into a new genetic family within the project. So my role as an administrator is to organize these projects, look at the new members coming in, and group them accordingly into Group 1, Group 2, Group 3, Group 4. Question at the back.
How unusual would it be to find a rare group like that with one person, and then the second person that ends up in that group is a 0 match at 111?
It would not be unusual to have new groups appearing all the time, to have somebody appearing as a very, very, very close match or an exact match to that person, to the first person. Again, it wouldn't be too unusual, because what a lot of people do when they do a YDNA test and the results come back, which basically say, you're all alone in the world. You know, they'll wait for a couple of months hoping that they'll actually get another close match, and then they'll say, well, to heck with this, I'm getting my brother tested, so at least we can be in a family together. And, you know, the two of them will come back as a very, very close match. So it's not unusual.
But this would be like an adoptee situation.
In an adoptee situation, it does happen, but it doesn't happen that frequently. Usually, with an adoptee, if you test, you'll get the kind of result that you saw when I showed you my matches, the 5 Gleesons and then 3 other people that are non-Gleesons. And it just is a signal for the adoptee that maybe you should be looking for somebody called Gleeson as your biological father. Does that correspond with anything you might have in your non-identifying information or any family lore that you might have heard from Great Aunt Mabel? You know, that kind of thing. So we have a question here and a question here.
Where does the DNA come from Thomas Gleeson in 1609?
Right, okay, very good question. Where does the DNA come from for Thomas Gleeson in 1609? We don't have his DNA at all. But what we do have is a paper trail going back to 1609. Thomas Gleeson, born in Cockfield in Suffolk, he came over to New England. He prospered, he had lots of children, they had lots of children, and now he has descendants all over the New England area. So what we're doing here is we're only testing modern day descendants, and we are, if you like, triangulating back onto the common ancestor in the 1700s, the 1600s, maybe even back into the 1500s if you're lucky. Of course, with a lot of Irish research, and certainly with everybody in North Tipperary group number 2, we don't have pedigrees that go back that far. Most people will go back to 1800, and then that's their brick wall. Group 1 is lucky because they have colonial US ancestry, and the records in New England are very good and have allowed a lot of the lines, a lot of the people in lineage 1 actually do have paper trail records that do go back all the way to the 1600s. A lot of them actually are still brick-walled in the 1800s, some go back into the 1700s, but there's a 2 to 3 generation gap that they have to try and figure out, okay, how do I get back from 1780 back to Thomas Gleeson in 1609? And that's where the paper-based records come out again as well. Question
Is
here.
there anything comparable to this table available now?
How do you mean comparable?
I mean, with it all lined up like this?
Oh yes,
This wasn't available anymore?
No, sure. This is the Surname Projects used to be hosted on 2 sites, Family Tree DNA and worldfamilies.net, and now everything is transferred over to Family Tree DNA. So if you actually look at any surname project on Family Tree DNA, you will actually be able to see this kind of table. It just doesn't have the pretty colors.
Okay.
Okay, so it's the pretty colors I wanted to show you because they very, very nicely indicate the genetic signature. The numbers will still be lined up. I'll show you an example of it later on. And what you'll be able to see is the individual mutations, and I'll explain that later. But it won't show you this nice colored pattern because the way that the worldfamilies.net website set it up was they had a very neat way of showing how the genetic patterns fell out of the larger genetic pattern for the overarching haplogroup, which would be R1b for a lot of people in Western Europe. I2 is another haplogroup that we have in Western Europe. A haplogroup is just a very, very broad group of people with a largely similar genetic signature. And as man left Africa, they were haplogroup A and B and E1B1A, and then they went over to China and became haplogroup O, over to Western Europe and became haplogroup R, went down to South America and became haplogroup Q. You know, so different people, as they migrated across the planet, developed different mutations which gave them a unique colored pattern, a unique genetic signature. And what we're seeing here is just how the Gleesons developed a slightly more unique genetic signature compared to the overall haplogroup for Western Europe. Question here?
Yeah, I did my brother's DNA, and I didn't do 111, I did 67 on down. And
Sure.
no matches until 25.
So you did your brother's YDNA and 67 markers were tested and you got no matches. Why does that happen? Because there is nobody else currently in the database that actually matches your brother at that level. And it could very well be that your brother has a very unique YDNA signature. He could come from one of those branches of the tree of mankind that is virtually extinct, and there's only very, very few representatives of that branch of the tree of mankind still alive today, and only one of them, your brother, has actually tested and put his YDNA in the database. So in your situation, it's going to be a waiting game where you actually just have to wait for somebody to join the database who happens to be a match, or if you really get hacked off, you get your cousin to do the test, and then he'll match your brother, and then you can be a genetic family again, yes.
Well, what about like at 25 I have matches? But I've written to a few of them before, and nobody writes back,
Yes, there are various levels that you can compare. You can 111 markers gives you a much more robust comparison if there is a match than say at a lower level, particularly when you get down to 25 and 12 markers. If you match somebody at 12 markers, yes, you have a common ancestor, and it's probably about 2,000 to 4,000 years ago. So you're not going to get back that far. It's the same with 25. If you have a match at 25, it probably indicates you do have a common ancestor, but it's going to be quite a while back, maybe 1,000, 2,000, 3,000 years ago. We have a question here.
When you submit your the YDNA to the site and you're looking at your analysis, and there are a number of matches, does the system tell you you really should consider joining these surname projects?
I wish they'd say that. I really wish they would give you an indication of which surname project you should join. It does kind of in a roundabout way. If you go to, there's a button that says join projects, and if you click on that, it gives you a list of suggestions. but it doesn't, the list is not as comprehensive as it should be. Project, because as well as surname projects, there are haplogroup projects, and haplogroup projects look at specific branches of the tree of mankind. And they will have, 6,000, 10,000 members, because you're going really quite far up the tree of mankind. For example, haplogroup R, which is one of the most common in Western Europe, that emerged around about somewhere between 25,000 and 35,000 years ago. So there's going to be lots of descendants from that particular branch of the tree of mankind, and joining the haplogroup helps those project administrators running that project to get an idea of how that group evolved over time and divided into different sub-branches. And I'll show you a little bit about that. I'm going to move on, but let's move on because we've got a lot to cover. It is, yeah. So we're going to cover a lot of the questions as we go through. But the important point is that DNA, the YDNA, can help group people together in similar genetic groups. It can help identify a person's origin, North Tipperary, West Clare, Suffolk in England, and it can even identify a person's named ancestor. It'll still be a challenge to get back the missing 2 to 3 generations to find that ancestor, but at least you know which direction to head in. And we've talked about human migrations, and I talked about haplogroup Q over here, haplogroup O in China, haplogroup R1b in Europe, haplogroups A and B in Africa. And associated with these human migrations is the tree of mankind and the various haplogroups. And as you come down the tree of mankind, you'll find that certain geographical areas become associated with certain DNA markers. Not only that, but those specific DNA markers are associated with certain surnames. And those certain surnames are associated with the names you find in those ancient historical texts. And in Ireland, our ancient historical texts, those ancient annals, go back 1,000 years or more. In Scotland, too, some of those pedigrees go way, way back. The Stuarts, the Campbells, all of those Scottish clans go back many, many, many generations. We're also coming down the tree of mankind into the surname era. And that's really quite an accomplishment when you think about it, because this tree of mankind started 250,000 years ago, and we're now at the point, certainly in the Gleeson DNA project, where we've come back down into the last 1,000 years when the Gleeson surname would have been around. Because surnames roughly arose about 1,000 years ago in Ireland, maybe about 800 years ago in England. Some parts of Wales only took on inherited surnames in the 1850s. So before that, they had a patronymic system. Eric, son of John, Johnson, William Johnson, Patrick Williamson, John Patrickson. It wasn't an inherited surname, it was a patronymic surname. But this is the sort of thing that ultimately you should be able to get in any surname project, is a family tree for each genetic group within the project, going back and showing you the evolution of the surname over the past 1,000 years. So, interpreting your YDNA results, I have an in-depth presentation on YouTube about this. So if you do want to go into depth about how you interpret your YDNA results, then this YouTube presentation is well worth looking at. You just simply Google my name, Maurice Leeson, YouTube, interpreting your YDNA results, and you'll get a very in-depth presentation on that. But let's look at your YDNA briefly. And here we have the Y chromosome. If we unravel it, we get the double helix that you see here. And we get then all of these bases. T, A, C, G are the 4 bases: guanine, cytosine, adenine, and thymine. And they bond to each other across the 2 strands of the double helix. And G always binds with C, A always binds with T. And that gives us 2 types of DNA marker. And this is the main take-home message. There are 2 types of DNA marker. One is the short tandem repeat, or STR, and that appears as a number. And the reason it appears as a number is because it's a repeat of 3 sequences, TAC, TAC, TAC. The value for this particular marker is 3. And that's how it appears in your DNA results. And that's what the numbers mean when you look at one of those surname projects and you look at the results page. The second type of marker is a SNP, a single nucleotide polymorphism, and that just means there's a substitution. Somewhere in the recent past, maybe this letter A was a G in the previous generation, but there was a mutation, minor mutation, and it changed to an A. And now the offspring has an A, and that mutation will be passed on to all of that offspring's offspring itself. And that's a way of actually marking different branches in the greater family tree for a genetic group. The numbers, the STR markers, here's an example. Let's look at marker DYS458. And the particular motif here is GAAA, and it's repeated n times. And the average number of repeats is 17. So you'd have GAAA 17 times in a row. So the value for that STR marker is 17. And that's how it'll be written in your results. But that's the average. And sometimes the mutation means that when it's copied from father to son, instead of copying 17, it only copies 16. Or it copies 18 by mistake. And that's how these STR mutations arise, and that's why you get a range of values for a particular STR marker. And you can see for this one, the repeat of 17 is the most common, and that makes it the average. But 16 occurs in 24% of people, 18 occurs in 15%, and it gets rarer as you go down to low levels of the repeat or up to high levels of the repeat. So that just explains what you're seeing when you're looking at these STR values. And here are these STR values. We have the marker's names up here, and then the value for that marker is 13, meaning whatever the sequence of letters is repeated 13 times. The next one along is 24. That means that it's repeated 24 times, and 14, 11, and so on. Now, usually, when these STR markers are passed on from father to son, there isn't a mutation, and they're passed on intact. So the son inherits exactly the same thing as his father. But every couple of generations, there is going to be a mutation. And that is why we are able to track those mutations back through time. The other thing that your YDNA results will tell you just on their own is perhaps your predicted haplogroup. You know, are you likely to come from China? Are you likely to come from Europe? Are you likely to come from Africa or South America? So it'll give you a crude estimate of your haplogroup, but that is best performed by, confirmed by SNP testing. The important thing to remember is that your STR signature is literally a string of numbers. 13, 24, 14, 11, 11, 14, and so on. That is your STR signature. It's just a string of numbers. The SNP marker, on the other hand, is usually a name. So like M222 would be the name for the SNP marker for Nile of the Nine Hostages, which is a very, very prominent Irish DNA signature. And we can write your SNP progression as a list of these SNP markers. And a SNP progression is really just a list of each of the markers that characterize each branching point on the tree of mankind from that major haplogroup that you belong to, haplogroup OR, for example, all the way down to your current terminal SNP, the SNP that you've currently tested, because you can do quite extensive SNP testing that brings you all the way down to the present day. You can test up to 85,000 of these SNPs on your Y chromosome with the big Y test. So let's look at those tests. STR marker tests I've mentioned are either 37 markers, 67 markers, or 111 markers. You can see the average cost there. And then the SNP markers can either be a single SNP test, a SNP pack, which is usually around about $120, and they contain about 140 SNP markers, or you can go for the big Y test, which tests up to 85,000 SNP markers. It's usually $650 now. It can come down to $350 in the sale. So it's not for the faint-hearted. These are expensive tests. And if you were to do the 111 markers and the big Y, it would cost you $650. So that's why the autosomal DNA test is the best one to start dipping your toe in the gene pool with. And this is really just for people who want to go down a much more advanced route and try and find out a lot about a particular surname in their family tree.
How would you know which SMP test to take?
There are several ways that you can predict which SNP test to take. Now, don't do a single SNP if you can avoid it. Either do a SNP pack or a big Y. But looking at your results, and if you look at this one here, for example, this person has one match at 67 markers. The genetic distance is 6 steps away from an exact match. They have one match called somebody called Malloy, whose earliest known ancestor is John Malloy from New Orleans. But they have done some kind of SNP testing because their terminal SNP is listed as L1308. So that predicts what SNP you may test positive for. You would then, presuming you're a Malloy, you'd join the Malloy Surname Project. You would ask the administrator, who happens to be me, what do you recommend in terms of doing SNP testing? And don't do any SNP testing until you've asked the administrator for advice, because that's what they're there for. They're all volunteers, they're all doing it for free, but they will have a better overview of the Surname Project than you will. Also, join haplogroup projects, and in this case you join haplogroup R, and ask the administrators there, what SNP testing would you recommend for my specific DNA results? So always ask for advice, because it saves you money at the end of the day. So here this person has one match at 67 markers, he has 2 matches at 37 markers, and you can click on the tip tool. This is the orange icon there, and that gives you a prediction, an estimate of how far back you can go to your common ancestor. Where does the common ancestor sit? And it's you should look at the midpoint estimate. Get the estimate as close to 50% as possible, and that will give you the best estimate of where the common ancestor sits between you and your match. There is a huge caveat here. These are crude. They have ranges that go plus or minus 200 years, plus or minus 300 years on either side of the estimate. I always compare it to a genealogist and a statistician meet each other at a cocktail party. And the statistician says, oh, you're going to be pleased, I started doing my family tree. And the genealogist says, oh, that's great, who have you started with? And the statistician says, well, my grandfather. And the genealogist says, well, when was he born? And the statistician says, well, I'm 95% confident that there's a 90% probability he was born sometime in the last 24 generations. So it's statistically accurate, but genealogically meaningless, really. So you have to be really, really careful with these estimates and make sure that they don't mislead you into thinking that this definitely has to be where our common ancestor was born. We want to know plus or minus 1 generation. They're telling us plus or minus 300 years. So they are extremely crude, and you have to be careful. When you do get these matches, you should definitely contact them and share family tree information with them. Try to find if there is a common ancestor somewhere in your respective genealogies, and that can help you break through a brick wall. And it certainly worked for me with my Spearans, because when I did my test, I didn't expect to be matching Spearings and Sperrans, but we all matched each other genetically. And one particular group that we matched had genealogical data that went back to Limerick in the 1600s. And that allowed me to go to the University of Limerick Library a couple of years ago, get these musty old boxes from the Earl of Dunraven's estate papers, open them up, and smell the parchment. And the first document I opened had my ancestor's name at the bottom of it as a witness to the transfer of tithes, and it was dated 1697. So you never know what you're going to find. Now, the admin of these surname projects will group the people together based on genetic distance. So if you're looking at the 37 marker level, it's anything up to 4 steps away from an exact match. They'll group people together. At 67 markers, it's 6 steps away, 6 out of 67, and at 111 markers, it's 10 out of 111. And they will also provide an analysis of the match's SNP markers as well. So those are the 2 main jobs that a project administrator will do. So, and that's where you see the genetic distance and also where you look for your terminal SNP. But let's see the bigger picture. And that's when you should join a project. And there's various projects that you can join. Surname projects we've talked about, haplogroup projects we've talked about. Geographic projects can also be really, really useful because the administrators there have a knowledge of the geography of the area that the YDNA comes from. And one great one in Ireland is the Munster Irish Project. And they're working not just with all the names from Munster and Southern Ireland, but they're also working with the ancient historical texts that discusses how those names originated. So they are a wealth of information. And if you have a surname that comes from Southern Ireland, definitely join the Munster Irish Project. You should also join the Ireland YDNA Project. There's also Scottish projects, British projects, and so on. There's also special projects. One that I really admire is the Scottish Prisoners Project, because after the Battle of Dunbar in around about 1650 or so, Cromwell frog-marched about 5,000 Scottish soldiers down to the cathedral in Durham. And 2,000 of them died along the way, and then they were locked up in the cathedral for a week, and another 1,000 of them died or so, and then the rest of them were shipped out to the West Indies, which included Maryland at that stage and probably New England as well, and we have documentary evidence of the arrival of 159 of them. And what John Cleary is trying to do with the Scottish Prisoners Project is trace the descendants of the Scottish prisoners who arrived in New England in 1650 or so. And you may have heard recently that they have been excavating around Durham Cathedral. They have found lots of skeletons. And those are probably the brothers who died of the people that survived that were shipped out. So the next step is, can we get DNA from those skeletons and link it to people living here today? So a fantastic project that is of great historical interest. So these are just the different types of project that you should consider joining. And again, everybody in these projects will be grouped, and then the results will be analyzed. The benefits of joining a DNA surname project: it helps identify where your surname came from, helps you connect with same surname cousins that could help you break through brick walls in your family tree. It places you on the human evolutionary tree, the tree of mankind. It can date your surname, potentially. It can characterize the evolution of your surname. Which branches migrated? Which branches went to Australia? Which branches went to America? It can define the branching structure of your genetic family and identify which people within the group sit on your sub-branch of the tree of mankind within that family. It can also help determine the deeper clan origins of your family. Is anybody related to Brian Boru? Is anybody L226? Anybody Nile of the Nine Hostages, M222? We have a few people here. Okay. So it opens up a whole area of history that you wouldn't normally have access to if you were just doing paper-based genealogy. And it may be tangential, and you know the way we tend to get distracted. Oh, that looks interesting. You're looking through newspapers, and you end up reading the ads from 1877. I didn't know they had a Turkish bath, and that type of thing. So it also contributes to the growing body of DNA research as well. And this is the crest of the wave of citizen science. This is all new stuff. This has only been around for the last 15 years. So by doing a YDNA test and being part of a project, you are actually helping advance science. And you are on the crest of the wave of scientific discovery.
Is anybody using all this stuff for medical implications?
No, because on YDNA there are no medical implications. Okay? Sometimes you might be able to predict whether somebody was infertile, but by the time they get round to using a YDNA test, they probably know that already, given the average age of a genealogist. So, you know, we're not looking at a huge amount of DNA, and certainly not a huge amount of medically relevant DNA with the YDNA test. 37 markers, 111 markers, it doesn't tell you a huge amount. But I'll give you a practical example from the YDNA project, just so that you get a sense of what analyzing your DNA can actually do. And I'm finishing in 10 minutes, okay. So, this is the results page of the YDNA project, and you can see that it's divided into 6 groups, and then the last one is ungrouped. These individuals here are the ones who have done the big Y test. And we can see that group 1 comes from Ireland. Group 2 is coming from Ireland as well. Group 3 from Ireland. Group 4, from Offaly. Everyone says Ireland, 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 but you'll see there's an Offaly there, there's an Offaly there, there's an Offaly there. There's 3 people from Offaly, and that's kind of pointing in an Offaly direction. Offaly is a county in the middle of Ireland. This is why I say it's so important to put the birth location of your most distant known ancestor in this box, because that helps us define where the group may have come from. Group number 5, well, they've got Scotland, Northern Ireland, Scotland, and then United States, Northern Ireland up there. So it could be a Scots-Irish group. So what's a Scots-Irish group called Malloy? Why is it called Malloy? And then group 6 is Ireland. But if you look through, you find that some people aren't called Malloy at all. Gamsby, Dempsey, Callahan, McSparran, Purcell, Macmillan. What's going on here? Why are we having different surnames? And the reason is there's either a surname or DNA switch. Alternatively, these surnames are you matching people that where the common ancestor is before surnames were introduced. And that's more than 1,000 years ago. Chances of that happening are actually very, very low. The other possibility is that these are chance matches. And just by chance, the string of numbers of one person's genetic signature happens to come fairly close to the string of numbers of somebody else's genetic signature. And that is known as convergence. It does happen, and it is one of the explanations for why you have people with different surnames in your list of matches. But let's take a look at we'll take a look at some of them later on. The last group is the ungrouped section. And why do they have no match? Either, like I say, they're a rare branch of the Malloy family and they're the first person in that branch to do the test. They'll have to wait for somebody else to come along. Alternatively, it's a surname or DNA switch. They're a Malloy by name, but not by DNA. There could be a Gleeson by DNA or a Murphy by DNA. There's some kind of switch has happened somewhere along that father line. And that's frequently due to a break-in transmission. We apologize for the break-in transmission. The surname or the DNA from one or both parents has not been passed on to their child. And there are a variety of reasons for why that might have happened. There's a disconnect between the YDNA and the surname. So, for example, this is the line of descent in the blue squares. But supposing this father here in 1765 abandons his family, what are the children going to do? Are they going to keep his name? Are they going to take the mother's name out of respect for the mother that raised them? In which case, the mother's name might be O'Flaherty, and that O'Flaherty name passes down along that direct male line, but it's associated with the YDNA of the husband who abandoned the family. And that's one reason for why you can get these surname or DNA switches. They are common. So this would be, illegitimacy would be another reason as well. The mother has a child out of wedlock. The child takes the mother's name on the birth certificate, but you don't know the name of the father. The father might be a Farrell, so it might be Farrell YDNA associated with the mother's surname. Another reason would be the interloper, as my father calls it. The postman always rings twice type of scenario. And a married woman has an affair with somebody from outside, Lady Chatterley's lover, and the the gardener's DNA, so he's got gardener YDNA associated with the name of the husband. So that's another example of a surname or DNA switch. They're also called non-paternity events, or not the parent expected, but I prefer the term surname or DNA switch. And it didn't just happen with illegitimacy or infidelity. There's a whole list of different causes. Swearing allegiance to the lord of the clan. I'm at Leeson, but the O'Briens are the powerful clans. I say, oh, Your Honour, I will change my name from Gleeson to O'Brien. And then all my children will, of course, be Gleeson by DNA, but O'Brien by surname. Surname or DNA switch. That happens with servants, soldiers, vassals, tenants, and slaves, all throughout ancient Ireland, from 1500 back to 1000 AD. Adoption, fostering, and guardianship, very big in Brehon society during the Gaelic clan times, and sometimes it would be associated with a surname switch. A young widow remarries, the children take the name of the new husband. You won't marry my daughter unless you change your name to Sidebottom. Okay, if I must, I shall. Taking the wife's name upon marriage. Oliver Cromwell was never Oliver Cromwell. He was Oliver Williams. But his wife's grandfather was Thomas Cromwell, who was the right-hand man of Henry VIII. So the family took the Cromwell name because it was of higher social status than the Williams name. So that was Oliver Williams. There was a very strange custom, apparently, in Celtic societies. Apparently Welsh was very common in Wales. It may have been common elsewhere as well, where if a powerful person came, the lord visiting, who he was visiting, would say, "Please sleep with my wife tonight, just so that you feel secure." And the chances that somebody would attack him in his sleep if the wife was sleeping in the bed were deemed to be relatively remote. Now, it was done for security and safety purposes. That's not all that happened, unfortunately, in the bed at times, which means that there is this other custom called naming, where the child on her deathbed would say, "Oh, darling, I need to have a word with you. What is it, my love? What can I do for you?" You said, "Well, you know our eldest." "Of course I know our eldest. Sure, isn't he my son?" "Ah, well, now that's what I want to talk to you about." So that apparently did happen from time to time, and it happened in the O'Neills as well. Hugh O'Neill was the son of a named child. But infidelity and illegitimacy under Brehon law was a very, very different thing to what we understand today. There was even stories that a woman could choose who she wanted to sire her child. So I'll have handsome Dave because he's big and he's muscular. But she could then decide who she wanted to raise the child. Because Dave is not very clever between the two ears, so I'm going to get Sam to be the father of my child, and he'll rear them, even though Dave has actually fathered them. So again, the past is an unknown country. These customs that happened 500, 1,000 years ago have resulted in surname or DNA switches to the extent that today you have a 50-50 chance that the surname you carry does not go back to the person that originated it 1,000 years ago. Because these kind of rates of surname or DNA switch are about 1 to 2% per generation. And there's about 30 generations back to the common ancestor, back to the start of surnames. So that gives you about a 50-50 chance that your surname does not go back to the person that originated it. But I always like to quote Shakespeare, and I think I'll end on this and then take a few questions. "A Malloy by any other name would smell as sweet." Romeo and Juliet, Act 2, Scene 2. A Malloy is a Malloy is a Malloy. A Gleeson is a Gleeson is a Gleeson. Whatever surname you happen to have, it's your surname. You own it. It belongs to you. And if we fall into the trap of saying, "Oh, well, my DNA isn't Gleeson," well, then you'll probably be wiping out 90% of your family tree research. We can't be that purist in regard to these DNA, surname and DNA switches. They happened all the time, and it's just the way that we are as humans. So I'm going to end it there, and hopefully it's given you some introduction to what YDNA can do for you. There's lots of YouTube videos as well if you search for my surname and look at some of the YouTube videos on the Malloy clan. The Farrell clan is the one that I put up recently as well, and you'll see how there's a lot of things you can do with surname projects, and I would certainly encourage you all to join one. So thanks very much for your kind attention. Now we have a few questions. I'll take this gentleman here and then this lady in the front.
As the only son of an only son of an only son going back 5 generations, does this help?
The only son of an only son of an only son going back 5 generations. This would be helpful because it will. This goes back to 1,000 years into surnames. It goes back 250,000 years into the tree of mankind. But presumably, and this is a kind of a caution with this YDNA testing, a lot of people who do YDNA testing, the real question they want answered is, can I push my brick wall back at least an extra generation? With YDNA testing, it usually isn't as simple as that. And you may have to do quite a lot of work to get that extra generation back. So if you have a brick wall on that only son, only son, only son line, it may very well help you push it back an extra generation, but it may not.
There'd be no real descendants.
Well, if you're the only son of an only son of an only son, it's not going to connect you with direct male line descendants until the there's an ancestor that had 2 sons, and one of them has had descendants on another branch. Lady here in the front.
Where are we going to find is it a video of today or is it just a audio?
No, it's this is being recorded on video, so you'll have these slides and you'll have the voiceover and you'll have the questions, and it'll be up on the website, which I think is the Tiara website or the Celtic Connections website. Celtic Connections website. Okay, so it'll be up there. I'll give it to the organisers and they'll sort it out. We have a lady here in the front.
How do you tell the difference between a non-paternal event and when the YDNAs are just kind of close to another family
Sure.
line?
How do you tell the difference between an NPE or a surname or DNA switch and when there's a false match because of convergence? There are various ways that you can do this, but a sure sign that you have convergence is if you open up your matches and there are 1,000 matches in your DNA results page. The majority of them will be false positive matches. There are certain branches of the tree of mankind that are prone to these false positive matches. One of them is M222, the genetic signature of Niall of the 9 Hostages, a warlord who operated in Ireland around about 500, 600 AD. What he did, he just took over the whole place and he eliminated the competition. In the way that they did back in those days, he killed everybody else so that his genetic signature became dominant throughout the country. And because it is a relatively recent founder effect, it means he had lots of descendants and they weren't that different from each other. So when they had descendants, they kind of crossed over a lot with each other, and that's where you get this kind of convergence of these string of STR numbers. But the best way of spotting it is to look at your results and see how many. If you have 600 to 1,000 at YDNA 37 or YDNA 67, good chance you have convergence. The way to kind of separate the wheat from the chaff is look at the terminal SNP. Determine your own terminal SNP and then see which ones are on branches that are inconsistent with yours. You'll find that a lot of your matches are on adjacent branches. Some of them might be on quite distant branches. They're obviously not closely related to you. They'll be related via a common ancestor 1,000 to 2,000 years ago. So SNP testing is a way of identifying convergence. Lady back here. Yes? Oh, a gentleman here, and then yourself.
How do they select the markers, like the 67 and the 111?
How do they select the markers? They have looked at a variety of DNA markers that are available because there's about 5,000 STR markers that are available. They look for the ones that go up and down quite frequently, the ones that actually mutate relatively frequently over the course of time. And that's going to give you a degree of power to distinguish between different genetic signatures. So they look at those that have a relatively good mutation rate, not too fast, but every 2 or 3 generations there might be a mutation there.
Without overkill, what do you suggest that which marker level do you suggest that we have done?
I would say start low, go slow. So you're going to start at 37 markers, and then depending on that, you're going to ask the administrator of the surname projects you join, the haplogroup projects you join, I've got my results back, what do you suggest that I do next? I'm thinking I should do this, this, and this. Would you agree? So you ask for advice. Don't waste your money on doing high-level tests without getting advice from a project administrator first. I'm going to be outside. I know that some of you have got other questions as well, because the next speakers are coming in. But thanks again for listening, and I'll be outside in the next few minutes.
Gleeson mixed in with a bunch of Collins matches from Cork, but I don't think you're one of them, from what?
That rings a bell, actually.